Okay. So Adrian and I were co-founders of Xsensio. I am the CEO. Adrian is CTO. He is also one key person in the company, in the strategy of the company. We're taking all the decisions together. And we have an advisory board, as I was starting to explain, with the Mayo Clinic is, as I mentioned, one of the shareholders of our company. Very interested in what we're doing. And we have the head of the ICU of the Mayo Clinic, one of the 3 sites that is actually advising us and officially a shareholder of the company. We have also a professor who is running our upcoming clinical study here in Switzerland from the ICU. And then we have the CTO of Texas Instruments. And I mentioned that Texas Instruments is a strategic partner. And we have actually an MOU with them, which is very promising for us because, as you probably know, Texas Instruments has been the partner of Abbott in the first generation of biochemical wearable devices. And what we're doing at Xsensio is expanding this concept of CGMs to other biomarkers. So we're very, very happy to have them on board. So what we're doing, the main problem that we're actually trying to solve is that there is not enough monitoring of patients around the world. So patients are not well monitored in the hospital, let alone at home. And as a result of this, patients in the hospital setting can evolve without not being properly monitored. So our goal is to enhance the monitoring of doctors and nurses by automating certain. So monitoring is already very well implemented in hospitals. You would know there are ECGs that are recording continuously what patients are, so the heart evolution of a patient dynamically. There is SpO2. So there are a number of devices in a hospital that are already monitoring and supplementing the work of doctors and nurses. However, everything is around physical data, so number of heartbeats and oxygen levels. What we are doing at Xsensio, we are actually changing the biochemical side. So right now, this industry has not really evolved much. It's still blood tests that we have to take, and it gives you just single point data every few hours or once per day. So what we want to do is go from single point to biomarkers in a wearable format. So tracking the biochemical side of patients the way physical data is already tracked in a continuous way with wearable devices. So this is the big market opportunity that we're going after. So how we do this, the Lab on Skin, which is AI patient monitoring system, it's a device plus AI. And what we do is a wearable devices that is put on patient that tracks continuously a number of biomarkers, and we're building a platform around it. And the very novel side is the whole concept. So it exists for glucose for diabetes and has transformed diabetes care. What we want to do at Xsensio is expand this to other biomarkers. So we're working on a pipeline with the first concept, the first application is almost ready to go into clinical study. So we had questions about the IP portfolio. We could dive quickly, but we can also go and just continue quickly the presentation. So mentioned that we have a platform that we're developing, and this platform has potentially many different applications. So glucose, we know it's one of the applications. So our goal is not to compete with Abbott, Dexcom, and Medtronic on this first application. Our goal is to do other things. So building other sensors to complement and go into multiplexing. So we have, each line is an application. So most of our work and most of our resources go to inflammation. So we want to be able to track inflammation continuously and help doctors identify when a patient starts to be really unwell. Today it's done by taking a blood test, looking at lactate and changing or adapting the treatment to be able to bring lactate down. So lactate is the single most important biomarker in the ICU. When it goes up, the patient is really unwell. What we want to do is with our Lab on Skin platform, we put a sensor for lactate that actually tracks lactate, not in blood, not single point, but in interstitial fluid and continuously. So it changes completely the way inflammation is monitored. So we have our first clinical study that you can see here. This is where we are devoting most of our resources today to build a small series of our device. The team has been testing it today on them. It's going to be going into clinical study in a few months on 140 patients inside the hospital. And our goal is really to help detect changes in lactate very quickly so that the doctor can be much more reactive and change the protocol very quickly, change the treatment very quickly. We have also in the pipeline cardiovascular biomarkers. We have also cancer. And so there are multiple applications. We're always starting in inpatient care. This is where the willingness to pay is the highest, but we see outpatient and home market as huge opportunities for the company. Yeah, so basically what we're doing when you compare with other solutions, so as I mentioned, the medtech industry has been working on physiological wearables. Abbott, Dexcom, Medtronic, with the revolution of CGMs, have changed that to be able to have wearables for biochemical data, only for glucose. And what we're working on is expanding to lactate, pH, CRP, NT-proBNP, and other biomarkers with a proprietary solution. What we could maybe just quickly go, because there was one of the questions that been asked was, what is the IP of the company? So what we have so far, so the device, we have 10 families of patents. Some come from the lab. Initially, it used to come from the nanotechnology lab, from which we are a spin-off of that lab. And this was mostly around the sensors themselves. And we got every time an exclusive license. But then more and more, the company has developed submitted their own patents, and then some have been granted. So we're extremely happy about that. We believe we have a strong IP protection, but however, we're actually devoting resources to continue to build really a very defensive protection around our IP. The patent portfolio is around multiple different things. So you could see here in this slide, this is our current IP portfolio. So some you could see Xsensio as the official owner, and some is EPFL, which is a Swiss Federal Institute of Technology, every time with an exclusive license to the company. Maybe we can just quickly talk about the upcoming filings.
Yeah, so in fact, these patents of EPFL are in a phase where the technology is, let's say, de-risked. And after de-risking the technology, we transfer it to Xsensio. And we have an official agreement for that was signed between my lab and Xsensio at EPFL level. And the family of patent that you see here, the patent at collection level, because the company started with this concept of collecting ISF through hollow needles. And then in the microfluidic layer, we have the sensor integrated that is sensing in very tiny amount, let's say 2 to 10 microliter. So we have patents that integrates the collection together with a microfluidic and with the sensor. And then we have patents on the methods of sensing that are specific to certain analytes. And there are patents now that will be more and more per type of product. And Esmeralda asked me to explain you the one that are for our first generation of product. Here we have 3 patents, one for which we applied last year, and the 2 that are now in advanced preparation to, let's say, to address 2 key inflammation biomarkers. The first one is lactate. So for the lactate, we have 2 key patents. They are not in the public domain. Again, as I said, one was recent, another one we are going to apply for it. So one of the patent was about how to integrate a semiconductor-based sensor that is produced in a semiconductor fab like the one of TI together with a needle that can penetrate a few millimeters under the skin and sense directly in ISF without any collection. So it's very important patent because we do what Abbott is doing, but with a completely different type of sensor, which is a semiconductor sensor, which is much more scaled, which has an integrated readout because it's semiconductor, so we can do the sensor and the readout, but it has also the potential for multiplexing. So there are three key advantages, again, compared to Abbott: multiplexing, co-integration of readout and sensor, and extreme miniaturization that we have in this. A second patent that is a patent for the inflammation marker lactate is a patent about the functional layer. So we have a particular stack of layer that was developed here in order to make a sensor robust and according to the spec that are coming from the medical doctor. To give you just a very simple idea, in the normal range, that is 1 to 2 millimolar, there are a number of sensors, not necessarily that are designed according to medical specs, but that have been developed. However, when you go into the needs of the ICU, there you have a high dynamic that can go 10x over the normal concentration that you have in blood or in ISF. In ISF, you have a certain dilution, but it's limited. So then we develop the sensor that is able to have a certain accuracy over a much higher range than whatever is available commercially today. So in order to do that, we develop a particular stack of sensitive layer that is based on enzymatic layer with certain type of, we call them diffusion layer, in order to ensure this much larger range and the robustness over up to 24 hours of measurement. So these are key specs that we are fulfilling according to what Mayo Clinic and CHUV Hospital in Lausanne ask us. So to have this sensor in the first deployment working for the nurse shift. So the minimum is 12 hours, but it can go up to 24 hours. And then, of course, the sensor can be further developed, optimized for exactly like the Abbott sensor to work up to a few weeks. But at this moment, the one that we are deploying is really following exactly this spec. So these are the two key lactate patents, and we have a third one. This is a very, very interesting one because it's addressing a marker that is 10,000 times more diluted than lactate, right? We are talking now, we are going from the millimolar, right, to 20 millimolar, but we are going now in the micromolar range. And there is a specific protein that was largely tested as a biomarker for instance, COVID patient, to see inflammation in COVID patient, is also used in septic patient. It's called C-reactive protein. To our knowledge, there is absolutely no product on the market that is able to measure C-reactive protein in ISF according to the specs of hospital. And here we leverage on more than 5-year work that has been done for C-reactive protein development of the sensor, also together with a consortium in which Xsensio was a subcontractor. And it was a 5 million euro project together with Charité in Berlin, and now is moving to a 11 million project that is again with Charité, but also with another partner in Paris, with Gustave Roussy. So C-reactive protein, we are able today to do this type of sensor. This will be generation number 2 of product, which will include the protein sensor for the first time in a wearable product. And for this, we have developed, and we have now the team has completed and validated in the complex matrix in ISF, a new method. I can just tell you that we call it AC method, like we are using not a continuous current, but an alternative current method, in which we are looking, let's say, at certain feature concerning the impedance of a functional layer. And for this, we have, I think, a very important patent that is just now being formulated in final phase and applied. So these are the 3 new patents. When this will be on the market, let's say released, and we can put it in our list, I think this will have also a strong impact on the overall value of the whole package of patent. And we are moving now with this marker that Esmeralda mentioned, the inflammation one, from the lab to the clinical studies, first with lactate and second with CRP. And by the way, I want to mention what is very interesting. Esmeralda said it, but maybe not enough. All our clinical studies, like the one is for lactate, but even the next one that will be for CRP, are not funded from the investor money, but they are funded through successful money raised from different type of European or national project, right? So the lactate one, we have a project by InnoSwiss in Switzerland that is fully funding just the clinical study.
Yeah.
And for the CRP, we have the European Commission that is funding 2 hospitals with few million each to have this study, clinical study, based on our patent. So this is, I think, an important dimension of the company impact, because we were able to convince funding agency to fund our clinical studies. I think that's enough. I can take more questions if you want, but it's already good information for you.
Yeah. Thank you so much, Adrian. There is I know that Manuel has a question. Please, I'm just going to allow you to talk. Go ahead, Manuel. Please ask your question.
Hi there. Thanks for joining. Let me join. Just to summarize quickly what the IP is, because the slide deck mentioned like kind of something on a chip. In one case, I think on the slide where there's like the stack going from the needle up till the hardware. So my question is, just to summarize whether I understood it right.
Yes.
If you go back, so you have a patent basically on where is it? Can you go back to where you have this stacked kind of parts of your technology?
Yeah, this one.
Yes, correct. So I understood you have this patents on this sensing microneedles, as well as some procedures to put these on a semiconductor. Is that correct?
Yes, I think we have patents on the microneedles and their integration as well with the microfluidics and with the readout electronics. So you are correct by saying that when you integrate it with on a chip, you will have it, so the integration with the readout. Now, what I want to tell you that in fact the patent family have two kinds. One is a patent family in which you have microfluidic, and this is what we have developed strategically. We have the largest number of patents that you see that Esmeralda cited in this table on this approach. So the idea there was that we are using a technique to extract interstitial fluid, and we are putting the sensor in the channel, in the microfluidic, very, very close to the needle, but not necessarily having the sensor on the tip of the needle inside the body. So here,
understood, yeah.
All this integration part, we have multiple patents for this. Why this is important? It's important because we believe, we are strongly convinced that for certain type of sensor, the integration can be done with let's say with collection, because we may have, for instance, some functional layer at certain moment that can be highly sensitive, but they are not necessarily all fully biocompatible, or they can go in point of care type of application. And this can be for very low concentration and you can use, for instance, aptamers, or you can use antibodies, and it's not always trivial to say that this integration is fully biocompatible. So this is one. Second technique. Whenever we are capable to have a functional layer that is accurate and sensitive, but is also 100% biocompatible, and it can go through the regulatory for biocompatibility, then we do not even think about having this collection. We have a technique now to place the semiconductor sensor in the tip of the needle in such a way, in this case, the stack, the functional stack is already a few millimeters under your skin and sensing directly into the ISF. This has a
Understood.
yes,
Understood.
so, okay, so this is exactly what but we have in our initial phase of the company, I would say probably 70-80% of the patent were on the configuration, sensor, microfluidic readout, system integration with semiconductor for the collection. But now we have the 3 new patents that I was citing to you that are, you know, this patent, for instance, the 2-1 for lactate, they are patents that are collectorless.
And maybe just say the value of this is by allowing us to get the sensors very, very deep inside a needle, we can be much more actually reactive. So the data, the sensing is very, very fast. And as you're tracking lactate and you're trying to save lives and getting the data as quickly as possible without a lag or reducing that lag to the minimum is really key.
Yeah,
understood. This is my last question quickly. What is the actual dimension, like physical dimension, that you're measuring from the electronics? You mentioned inductivity to some degree, but kind of at the end, is it some current or some frequency response, or what is it that you measure?
Yeah, it depends. I think we have multiple type of way to measure. The one that is for lactate is an amperometric measurement. Then we are measuring current, and we are measuring current, I can tell you, within the range of 100 of nanoamps and even sub 100 of nanoamp with a resolution that is 1 to 2 nanoamp, right, in order to reach the specification. But we have also voltammetric sensor. You know, you can imagine sensor then like ion sensitive FET. You know, this is a technology that has been established, is used only for pH sensing. And this ion sensitive FET have a measurement in which we can look at a shift of certain voltages. So we have also this type, and now the last thing, specifically for the protein, then specifically for the protein, we are looking at a measurement, exactly as you describe, is the we apply a signal that has a certain frequency, very specific to the detection of the respective protein. And we are looking at okay, it will be very technical what I'm going to say. We are looking
it's fine if you could just
the change
high level,
of the
it's fine for
phase,
me.
right? We are looking at the change of the
Understood.
phase. Right,
Understood.
so is the imaginary part.
But
this,
all fine. Yeah.
This is very, very common because you may know that in electronics, sensing capacitive variation is a very advanced technique, and that now we apply into the bio domain, right? In the
Understood. Understood. But I mean, my kind of synthesis to this is that you kind of build or have patterns around this kind of interface into the human, and less about the electronics that help you measuring this, because this is just kind of off the shelf stuff. I mean,
no, I would say is that some more standard, for instance, for the pH, maybe it was, or for lactate, maybe the way is more standard. But I'm telling you, for instance, the method for the protein is a patent that includes the method and the way you sense this, including the interface of electronics. So the last patent that I just mentioned for the protein, it includes as well the electronic part.
Yeah.
Okay, perfect. Thanks for the clarification. It's very interesting. Thank you very much.
Thank you, Manuel. There's Heikel also had a question. Please go ahead. Let me just allow you to talk. Okay, please go ahead. Ask your question. Let me ask you to unmute, Heikel. Yeah. Heikel, please go ahead. Yes, we can hear you now. Yes, go ahead.
All right. Yeah, thanks, Esmeralda and Adrian. My question is a follow-up on the previous one with respect to IP and competition. Assuming a competitor would use the same principle with discrete electronics instead of integration of a chip with with the needles in a monolithic way as you're presenting. Is this something that would lead to similar results to the product? And is it would this circumvent your IP?
Very good question. I will answer this way. If you stay at a certain range of marker concentration, then you don't maybe need, and a single marker, individual marker, maybe I can state that the results of using a coated metal needle, like like is used by this company now, that is a very simple method to just coat a conductor with a functional layer, and our semiconductor sensor will lead with similar results. However, now the more you look into diluted marker, and the more you will find that in order to track certain specific disease, you need what is called multiplex sensor. So you need to sense more than one analyte, at a moment of time. You have to multiplex. The more you dig into that, the highest our differentiation and advantages are. And this is why Esmeralda was telling you, if you start, in fact, we can do glucose like Abbott, we can do it today, but it was voluntary for us not to put a company in a competition for glucose. So today I can do here with the team, with Esmeralda in a person, in vivo, I can do glucose, lactate, and pH, all the three together, right? What Abbott is doing, we can integrate three in the same time with same precision, accuracy as Abbott, right? 10 to 20%. And this we can do. But now when you move into this type of highly diluted marker and the need of multiplexing, then you have all the advantages of semiconductor sensor, right? So you can do multiplex, as you know very well, Michael, because you've been in this field, if I remember correctly, right? You can have there on a semiconductor scaled number of electrode, even sharing a common reference electrode, multiple type of functional layer that can sense multiple analytes. And this is a gold mine in the future for specific disease for the medical doctor.
It allows also better triaging in the hospital. If you follow just one biomarker, then you cannot differentiate between different conditions and different situations.
Yes, for risk stratification
Risk
as well.
stratification.
Risk stratification in hospital is absolutely key. And of course, our decision as company, because we are small, right, was to say, look, we are not going to put in our product the maximum of complexity. We are going to have our first product that is designed for a marker in which this marker is in the clinical practice, and at better cost, we are going to offer the dynamic of the marker without doing the blood test. And then this is going to qualify our platform, medical grade. We are going to have this done at 2 sites, one now in Europe, and we plan to do it in US. And then we are going to fully leverage the other. But we have on the pipeline marker with different maturity, and the one that I cited to you now, including the C-reactive protein, they are now fully validated, and they are just waiting to be scaled up in the clinical study. And we have other marker. I can tell you, I have not talked today, but we have a patent and 2 other families of patent following on hormone detection. Again, this is something that will come
on the lab side.
On the lab side, that is being transferred now to the company, because again, as I mentioned, how people are working here, they risk completely technological sensor, and then they move it in the platform of integration system level into the company. So this is an open bracket. So just to understand how we are working, I'm not sure if I answer the advantage of semiconductor compared to the technique that I use in the industry.
Yes, Adrian, you did. Thank you. So for your, I understand what you're saying. For the first product that you're going to launch, the go-to-market will use only lactate.
Lactate pH.
Lactate and pH, because pH we can integrate it almost for, let's call it like that, for free. And pH is very important because sometimes people that are going in the ICU in emergency unit, they are unbalanced in electrolytes, and they have a pH pH is very crucial in a narrow range for the patient. So ask the medical doctor, I say, wow, it would be amazing if you can offer us on top of lactate, that is the most dynamic marker, another check for risk stratification, which is pH. So first product will do lactate and pH. It will be already a multiplex between a high dynamic marker and a very basic marker that is pH, however, very important to have it in the hospital.
Yeah.
Okay, thank you. Thank you, Adrian.
Great, thank you. I actually have one question as well.
Yes.
And which is to say, these microneedles, the way that they're able to sense from the interstitial fluid, of course, they have to sample it. And then there has to be some chemicals or some way of maybe some binding agents or something like that, which figures out what is it that you're finding in the ISF, right?
Yeah.
So what are those, and do you have any IP on that as well?
Mm-hmm. So let me comment on that. Yes, I think what you are referring to, for instance, there are particular type of sensing certain marker in which we need to have a specific sensing that is happening through binding, for instance, using an aptamer or antibody on fragments of antibody. This is exactly the principle we are using for the protein sensing. And again, in this case, what we are essentially doing in the company is that we have in our team a sub-team that is working first in validating the binding process through existing type of standard optical method like SPR, surface plasmon resonance. So we are making our own assay that is part of the development, that is they are using state-of-the-art equipment, and this is the whole advantage of the company being on the EPFL. EPFL is the university campus, because in life science department we have access to all this huge and very expensive equipment in which we are validating the assays that are based on the binding and debinding. So in some cases we are thinking about sensor that first we are looking at the process of having specific assay, non-specific assay, and using a subtraction technique from what you describe as binding. And yes, we are absolutely aware, and I think this is what you just described would be crucial for the protein sensing.
So we're not developing functional layers. I think the know-how of the company is finding the best functional layer among enzymes or aptamers, antibodies, or
Commercially available.
Commercially available. So finding that and then putting that on top of the sensors. That's the know-how of the company.
Yes, the biochemistry, how to integrate the sensitive layer into the type of device or electrode that we have. This is our know-how. But in principle, we are looking for existing aptamer and specific other type, enzymes or specific, for certain type of biomarker that we are looking at. Of course, at a certain moment, we may go, as the company is growing, to have subcontract to develop specific type of, aptamer with other company. This is something that we also started 2 years ago for a specific development of an aptamer with a company that is in North America. But we are very aware about this. We are working on the development in early stage of the assay, exactly as Esmeralda say, based on commercially available.
Okay, got it. Thank you. So just so I understand it correctly, I'm going to paraphrase what I understand. Is that, as an example, you draw blood and run a test, you're going to run the test in exactly the same way, or more or less the same, or you're testing for the same things as you would through an Xsensio lab on skin patch.
Yeah, maybe explain that it's a label-free method. So there is no, I mean, in the centralized labs, or you would have all the mixing that is happening, and here this is not happening. So
it's label-free. We are not using an amplification here if we want to test. So in fact, well, to tell you something very important, the company we are also running, we have run different internal studies that are super valuable, and this is know-how that is not necessarily something that we patented, but is kind of internal secret, I will call it like that, about the content of ISF in proteins, the dilution, and the correlation with blood. This is very important because, the marker on which we are working now that are in our pipeline, there are markers for which we have the proof that they are both in blood and ISF, and there is significant correlation between the concentration and or the dynamics. And this is very important. You will have some literature in the domain, but not for all of them. And because of that, we are beyond the literature. We have our own internal studies that were run through with let's say, according to regulation that are in Switzerland. And we had at least 2 studies that we run internally on the composition of ISF. I can tell you one that it was for us amazing. Was we have for CRP a study that was, we took the opportunity of COVID period in which people are getting vaccination. After the vaccination, is a reaction of, you have an inflammatory reaction, and the CRP, C-reactive protein, is picking up in the first 48 hours after the vaccination, and then is going back down. And then this gives us the opportunity for people that were volunteer to make a study, CRP in blood, CRP in ISF, dynamics in both. And this put us in strong confidence to launch the company and the second generation of product on the CRP, because is correlated and it's very slightly diluted. So we are detecting now in the 1 to 10 micromolar CRP, which is exactly the most relevant range for the medical application.
Got it. Thank you very much.
Maybe I can just go over one other question that always comes up. It was the exit strategy of the company. So there is always this question that is coming up. So maybe we can just quickly, let me just go. So the goal of the company, at least at the beginning, is to sell the product to hospitals. The hospitals are going to be our customers. They are responsible in actually making sure the patients are safe, and they are reimbursed for every patient. Now, in the US and in Europe, hospitals need to manage the cost in the best way. So we believe, and talking to many hospitals, our tool is going to be a tool that helps hospitals make better decisions and prevent risks from coming up. So by detecting the onset of an inflammation, a hospital would be able to intervene faster, not having patients coming too late to the ICU and actually overcrowding the hospital, overcrowding the ICU. So it's very costly today for a hospital to have a patient that is at septic shock plus organ dysfunction. So our goal is to prevent that from happening. Now, in terms of exit strategy, we see different groups of corporates that could be super interested, and we are already in discussion with a few. So, for example, companies like Philips and Siemens are already selling to the ICU, already having all these sales channels to the hospitals. What they're selling is not what we're doing. It's not wearable for biochemical data. It's wearables or point-of-care devices, traditional. They're interested. They see the market shifting. They see that you need to be closer and closer to patients inside the hospital to better track what is happening, but also follow the patients when the patient is released back at home. So this is one category. As Adrian has talked also a lot about Abbott and Dexcom and Medtronic, we see also an opportunity where our technology could be really useful and very attractive to these companies that have CGMs, that have poured a lot of money into CGMs, and the market for CGMs starts to be really standardized and commoditized. I mean, they're all selling solutions that are very similar, and they're just competing on expanding the number of days, and they're all looking for something new. And as Adrian was explaining, our solution would enable them to really open up a whole new world where you can multiplex with highly miniaturized sensors. So this is a second category. So for both medtech products that are already in the hospital, we are complementing potentially their offering. And for companies that are already in the biochemical side, CGM companies, we are expanding the number of biomarkers, so expanding the platform by going into semiconductors. So I just wanted to quickly go over this question that always comes up.
Sorry. But Esmeralda, I guess one of the questions also is, when you have an exit, will it be to the Siemens and GE Healthcare of the world? Is that what we're looking at? And if so, what are the comparables in this space that have, comparable acquisition transactions that have happened?
So the market is still very, very fragmented. So there are a lot of that there are a few big players that we believe are really operating in a very commoditized market. So it's very hard for them to differentiate themselves. So it could be on Siemens, GE Healthcare, Philips that are selling devices to hospitals, or more and more so on the CGM side, which is much closer to what we're doing. Abbott, Dexcom, Medtronic that are also in a very competitive market right now. So we believe these two segments could be very find Xsensio very attractive, potentially even more on the CGM side, because we are enabling them to, by integrating semiconductors, open up to many other biomarkers. Right now, I mean, lactate could be a sort of a natural extension to what they're doing, but proteins and hormones that are also in the lab of Adrian could be, I mean, probably not really achievable with the current solutions of Abbott, Medtronic, and Dexcom.
Okay, got it. Thank you. But so we are looking at these two as potential exit options. Okay, understood. And how long, like in an exit scenario setting, we always need to understand, okay, how long, how many years is it going to take? And are there comparable transactions that have happened? What was the size of those transactions?
Yeah, so the comparables that have been happening are really on the conventional medtech solutions. So not really on the wearables yet. So wearables for biochemical data, not yet. So it's a very, very novel market, and we are pioneering this market. There's been a few very big scale exits that have happened around vital signs monitoring. That is a market that is really starting to be mature. In terms of timeline, I think we're looking at different windows of opportunities. A natural one would be probably when we have FDA approval. I don't think we need to really build a huge sales force and go into that. I think it's the value of big corporates that could do that.
Maybe to be more concrete, I think for the CGM, that is kind of more than, a number of years ago, before these three bigs were in the company, it was an exit for the glucose of a UK Swedish company of more than 1 billion.
Yeah.
Right? If you want a concrete example. So the CGM that is now on the market, it has initially in the pipeline a startup that exited, and it was acquired by one of the three bigs of more than 1 billion exit.
By Abbott, yeah.
By Abbott, just the glucose, right? Without capability of being multiplexed. And glucose is now a 6 to 9 billion market per year, and probably extended. And I think we are very privileged the fact that the company that did all the integration with electronics and the readout for the glucose, which is TI, is the company that is working with us to do the specific type of readout integration. Heikel asked me that. All this integration now, we are working with this company. And this is the reason we are putting our IP and the main IP, not necessarily an inventing new circuit, because this is the business of other, but it's really on the type of functional layer and integration of functional layer with the semiconductor and multiplexing and the method to sense that. So this is how we distinguish ourselves here.
Thank you. Heikel has another question. Please go ahead and ask it, I think.
Yeah. Right. So, Esmeralda, it's more probably to you with respect to what you said last. So the ISF measurement compared to blood measurement, how representative is it? Is there a delay or anything that may make the measurement work a different way that we need to be aware of? Does it have to be validated?
So it has to be validated. So the clinical study that is going to happen at TÜV in a few months on 140 patients in the hospital is going to be really not changing the protocol of doctors. They're going to take blood tests the way they do today. And then in the background, we're going to collect continuously with lactate in ISF. And then we're going to show, it has been proven in the literature already, but we need to show it, that we are able to see the onset of changes and we have the same dynamics. Now, by having continuous, we will be able to demonstrate that we have a lot of value in doing continuously, because we can see the onset upward and downward.
But again, there are validation and there are a number of significant paper and data in the literature. We have also run internally by running specific tests on healthy volunteer in which you make intense exercise. In intense exercise, they are going to generate lactate in your muscles, and then you are measure it at systemic level, and you take sample of blood, and in the same time you measure lactate, and the people have seen, we have this data if you internally, but again, we can also show you literature. So for lactate, I think we are at, I would say, if not, maybe I would not say no risk, but it's almost no risk because it's known absolutely clearly that there is a correlation, and we are even running into a medical hypothesis that is made by, maybe you explain.
Yeah, so the ICU doctor believe that our tool is likely to be a precursor of changes of blood. So we would potentially be able to see the changes before they are even visible in blood. And the assumption is that lactate is a measure of tissue hypoxia. Since we're measuring in the tissue, we'll be able to see that change happening before it goes into the bloodstream,
into the systemic level.
Yeah.
So which means we can be, to answer your question, for a particular case of lactate, we can be even faster than the measurement in blood to do the risk stratification. So I believe we believe, because this opinion is shared by at least 3 major clinics in the world, right? Is University Hospital in Lausanne, Charité in Berlin, and Mayo.
Yeah.
So then this is our model, to work at least with 2 medical sites each time. So the 3 are totally convinced that our first product is not running in any risk of correlation or dynamics. And as we are measuring without collection, practically we remove, you are right with something, that is the delay of collection, right? Yeah. There are some companies that are collecting, the collection may take 15 minutes, and then you see a lag.
Yeah.
But in our case, this is not anymore existing because we are using a sensor that is using the same principle as Abbott without collection for the lactate. Yeah. So we eliminated practically the lag of collection from the high dynamic marker like lactate. For other type of markers, like in CRP is changing much slower. So in this case, we can tolerate a collection lag of 10-15 minutes, right?
Yeah.
So this is the time needed in order to have 2.5 microliter in which we measure. So depending on the dynamic of a marker, you may tolerate or not a lag of collection. Lactate dynamic, no, we are not tolerating. We are having a sensor solution without collector. On other marker, maybe we can have solution with a collection that are really adapted for this. So we are very, very aware about the marker that have low and high dynamics. And we try to adapt, our solution to be the simplest, the cheapest. And, we don't need a very complex solution when we can answer a certain need with a simpler, cheaper, and straightforward, robust solution. So I learned also this myself, because sometime in university, in research, we want the best, the most extraordinary sensor and so on. But no, here we are very pragmatic in what we are doing.
Yeah.
Thank you. Thank you, Edwin and Esmeralda.
Great. Great. Thank you so much, Esmeralda. We can wrap up now, unless you have other stuff to do.
No, I think we've covered the main questions that we get a lot.
Okay, fantastic. Well, thank you so much, Esmeralda. Thank you, Adrian. This was fantastic.
Thank you. Hope it was useful.
Okay. All right.
Thank you very much, everyone. Bye-bye.
Bye-bye.
By mail, we can answer other questions in case there are others. Thank you.
Yes, by all means, we'll send them to you.
Bye-bye. Bye-bye.
Bye-bye.